Hello, this is Mr. Watkins. Welcome to another video lecture. This is going to be on the light reaction. This diagram can be downloaded from the website. So let's get started here. The light reaction occurs in the thylakoid. So this is a thylakoid membrane. This is the inside of the thylakoid. And out here, this is the stroma, the solution that surrounds the outside of the thylakoid but is inside that inner membrane of the chloroplast. So let's get started here. We have both P680 and P700, photosystem 680 and photosystem 700, or also photosystem 2 and photosystem 1. Together they form a photosynthetic unit. These work together to convert light energy into chemical energy, and in this particular case, this electron transport system here does that conversion to NADPH2. So I'm going to focus here on P680 and walk through, but please note this chlorophyll A molecule right here, and this same chlorophyll A molecule, both these two molecules are involved with the scenario of having light hit their double bonds and knocking an electron off. So we'll just start here at P680. So light hits the plant. It can knock off an electron. That electron would absorb this energy, become more excited, and leave the chlorophyll A. These are antenna pigments. So at different wavelengths of light, these antenna pigments will do the same thing. They'll just send their electron to chlorophyll A. Chlorophyll A then removes that electron and sends it on to this special molecule called a hydrogen carrier. A hydrogen carrier is also called a proton pump, and we'll talk about that in just a moment. So the chlorophyll A gives up its electron, becomes oxidized. The hydrogen carrier picks up the electron and becomes reduced. Picking up this electron increases its energy. It carries the electron from this P680 to this whole unit, this photosystem P700. In the process of going from P680 to P700, the hydrogen carrier or proton pump will pick up hydrogen ions out in the stroma. These hydrogen ions, as the hydrogen carrier moves, gets basically pumped against the concentration gradient to inside the thylakoid. This is going to be an active transport using a carrier protein. In this case, we call it a hydrogen carrier or proton pump, pumping or moving these hydrogen ions against the concentration gradient inside. So we're going to build up potential energy on the inside by building up a difference, a higher concentration inside. Once the hydrogen carrier gets to the other side, it's already released its protons. It's going to release its electron to P700. When it does, that hydrogen carrier is oxidized, P700 is reduced. And again, it goes through a bunch of different molecules here, including chlorophyll A, and ends up on this last molecule called FAD. FAD picks up that electron, becomes reduced, and then uses that to store that electric energy, that chemical energy, in a molecule called NADP. And NADP plus some hydrogens and the electron that was trapped by chlorophyll A, that light trapped that energy into that electron, move these electrons, move that energy through the system. It will get stored in NADP and some hydrogen ions into a molecule called NADPH2. This molecule will be used then in the dark reaction of photosynthesis, or the light-independent reaction. So it will go on to another process that we'll have a lecture on later. Now, in this process, you need to note that we do have times when molecules are oxidized, lose electrons, or reduced, gain electrons. When you lose an electron, you're going to lose some energy. When you gain the electron, you'll be gaining some energy. Energy is not lost or destroyed. We're just transforming that energy from light energy to chemical energy and then storing it in molecules that can be used. Now, if this if this chlorophyll gives up an electron, then it is oxidized, and for it to work again, it's going to have to be reduced. It's going to have to gain electrons back. And that process by which this chlorophyll A gains that electron back, or gets that electron back, is called photolysis. Now, photolysis is light splitting. Basically, the water is split into three different pieces. Those three different pieces are: 1, electrons; And again, those electrons are going to be used to replace the electrons lost in chlorophyll A. It is also broken into hydrogen ions or protons. And those hydrogen ions are going to join the hydrogen ions that got moved across the membrane by the carrier molecule. And so we have got hydrogen ions being dumped inside by the carrier, hydrogen carrier. We've got hydrogen ions being dumped inside through the process of photolysis, that splitting of water. So we have a big increase in hydrogen ions here. The other piece is going to be oxygen. Oxygen will be given off, and this is the waste. This is where we get the oxygen that we say in the equation for photosynthesis. This is the oxygen gas that's given off because water has been split. Now let's talk a little more about all these hydrogen ions. As the hydrogen carrier molecule pumps these hydrogen ions to the inside, as photolysis continues to split water, creating hydrogen ions, we build up a concentration of hydrogen ions inside the thylakoid. That concentration is reduced, or at least the pressure of having all those hydrogen ions on the inside is reduced by this special molecule here, and we're going to call that ATP synthase. And this molecule is a channel protein. And the process that we see here is facilitated diffusion. And what happens is we're going with the concentration gradient. The hydrogen ions go through ATP synthase. There's a special molecule in the top here. You don't need to worry about what that molecule is or how it functions at this point for PreAP. But what you do need to understand is the hydrogen ions go through this facilitated diffusion through this ATP synthase, and in that process of moving through, that energy is used to change ADP to a molecule ATP, and energy then being stored. So there's really two places in the light reaction where energy is stored. There's one place where we have the conversion, and that conversion is in the chlorophyll A or the antenna pigments, where the light strikes electrons in a double bond, excites them, they bring up the energy level, and that electron then is knocked off, literally jumps off of its orbital. That's where the light gets converted to chemical energy. And then the rest of the process is just transferring that energy to the molecule that is needed or will be needed in the dark reaction, NADPH2. Part of the process then also, because of the movement of this electron, allows the cell to use that energy, that movement, to take hydrogen ions and move those hydrogen ions to the inside, as well as using photolysis to create more hydrogen ions to increase the concentration inside the thylakoid, using that natural movement from a high concentration to a low concentration, or facilitated diffusion through a channel protein to store more energy in ATP. Now please note, these hydrogen ions that are out here in the stroma can go back through this whole system, and you basically get a cyclic effect. Now the two energy storage molecules that are here and here, both of these two molecules will be used in the light independent reaction. I want to recap the reaction, the light reaction, so that you can get it in a format that you may have seen in a molecular format here. So we've got our light reaction. We need, for the light reaction, we need water for the photolysis. We need light energy, and we need chlorophyll A. At a minimum, chlorophyll A. Any of the antenna pigments would basically work, but chlorophyll A. And what we get from that reaction then, from water and the light energy in this complex that has chlorophyll A in it, we're going to get ATP. So we're going to store energy in ATP. We're going to get NADPH2. So we're going to store energy, that energy from the light, into NADPH2. And we're going to get some oxygen on the end, and this will be given off as a gas, as a waste product. The light reaction, energy is not created nor destroyed. Energy is transferred from light energy and stored as chemical energy in these two molecules. In the next lecture, you're going to see that these two molecules are then used to build a sugar molecule, taking carbon dioxide and basically transferring the matter into just a simple carbon dioxide to a glucose molecule. Thank you.